Hi, class. So now what we're going to look at is the different aspects of a protein. Right? So when we talked about protein structures, we said that there's a primary structure, secondary, tertiary, and quaternary. And so now I just want to use the video to give us a little time to really talk about each one of these in more depth. So the first one we're going to talk about is the primary structure. A lot of times you'll see me just kind of put a 1 with a little degree sign on it to talk about it. What this is the sequence of amino acids. All right, so if we're looking at this one, now I'm going to be lazy and just write AA for amino acids. What it's just telling you is what's the order of amino acids. So what amino acid goes first, which one goes second, and so if we have a long protein chain, which one goes first, second, third, and so forth. All right. So all the primary structure is talking about is what's the order of amino acids. All right. So if we started cutting off the pieces at the end, which one would come first, then second, then third. All right. The next one, and let me change colors just so we can kind of make it easier to keep track of everything. The secondary structure is what a lot of times we call the local structure. And the reason we call it the local structure is we can actually have a handful of these throughout the protein, right? And they can be different, they can be the same. Some of the most common ones that you'll see is the alpha helix. The alpha helix is one of the more common ones. Sorry, let me clear this up. That's my symbol for alpha. And then helix. And with the alpha helix, it's going to look kind of like a spiral staircase. And that's the one that's shown here. If you can kind of see this kind of spiral staircase where it's kind of rotating around, it makes these really nice cylindrical structures. All right, the other one that we'll see is beta sheets. And we'll talk about both these structures in a little bit in more depth and why they're stable and why they're good structures and why we tend to see them and what are the different effects of each of them. But what you guys should take home from this video is the secondary structure really refers to what we call the local structure. And those are typically your alpha helix and beta sheets. And there's a few others, but those are the two that we'll look at most in this class and are usually the most prevalent. So the alpha helix and beta sheet is what you should consider with the secondary structure. All right, the third one, and we'll look at this in a little bit more detail here in a minute, but is the tertiary structure looks at the way the protein folds overall. So the way this group folds. And so if you can see, this green line right here in the inside is trying to tell you, well, maybe this strand here is an alpha helix. All right, so we have an alpha helix in this part. But then over here we may have some beta sheets, and here's some more alpha helix. The tertiary is really about the overall (3D) shape. So how does it fold together? All right, so how does it fold together? How do we put all these pieces, the primary, the secondary structure, how do we put them all together into, it looks like a big blob, but it's actually really highly ordered. All right, the last one, and the quaternary isn't always clear on how it's different than the tertiary, but what it is we're going to talk about these subunits. And it's when these different units come together. So we take a tertiary structure, which would be one of these yellow ones, and we put it together with some other units. How do all the pieces come together? And so I think that's kind of the best way to look at it is when we're looking at the quaternary structure, how do all the pieces come together? And so usually by those pieces, what we're talking about are the subunits. Right? And so how do these different groups here and here come together? right? And sometimes they're multiple of the same ones. They can be different ones. They can all be different. But how do all these pieces come together? All right? And so what I would do is commit to memory the difference between each of these. All right? And if we recap briefly, the primary sequence is the sequence of amino acids. So what amino acid comes first, second, third, fourth. The secondary structure are things like the alpha helix, beta sheets. And you're going to see you'll have multiple of these in each enzyme once it's folded correctly, or protein. The tertiary structure is how all that folding comes together and what does the three-dimensional shape look like. And then our quaternary structure is how do we start to put those pieces together and get this big overall shape. All right, I hope this was helpful.